Hello, everyone. Good afternoon. This past year, my partner and I have worked to develop a prototype of a 3D printed device that detects Staphylococcus aureus in patients in under 30 seconds using volatile organic compound metabolites and specific enzymes. S. aureus is a very common bacteria, and it is the second most common healthcare-associated infectious bacteria. Pathogenic strains of S. aureus can be potentially very deadly, as hospitalized mortality rates reach up to almost 30% in many developing nations. A case of pathogenic S. aureus can result in many respiratory infections, as the bacteria spreads primarily through respiratory droplet transmission. The current diagnostic procedures, such as swabbing and then incubating the bacteria, can take up to 48 hours and could cost hundreds of dollars to the patient, creating a need for rapid, low-cost diagnostic options. There are some current rapid diagnostic treatments, such as polymerase chain reaction and rapid staph test. PCR finds a genetic biomarker found in S. aureus and amplifies it, but this method is not widely available and can be expensive. The rapid staph test shows a line where samples are put against a nitrocellulose film, or it shows a latex mask where antibodies are present in the patient. But these tests are not very accessible to people in developing nations, and they are not self-administrable. Our test we developed includes 4 biochemical tests containing 2 droplet-based breath tests and 2 direct contact tests that consist of applying a sterile swab in the patient and placing the swab in the associated test solution. We chose this model for the test to limit the amount of equipment for the user, and so it's cost-effective and easily used. Our first test we have is our calcium hydroxide test, where the patient can make sure their results are correct and based on their breath compared to outside contaminants. When users blow into the device, the 4 to 5% of carbon dioxide that is in their breath will react with the calcium hydroxide solution inside the device to make carbonic acid. When you put a colorless pH indicator like phenolphthalein into the test solution, the solution will turn from pink to clear as the basic calcium hydroxide morphs into the acidic carbonic acid. Our next test was a direct contact test, which is the hyaluronidase test. Many organisms have the hyaluronidase enzyme, but only these 3 bacteria contain this enzyme in the human respiratory tract. Our test comprised of hyaluronic acid and Benedict solution, which is a mixture of sodium carbonate, sodium citrate, copper sulfate pentahydrate. As the hyaluronic acid mingles with the hyaluronidase in the sample, there will be an oleosaccharide produced. Benedict solution detects the presence of simple sugars such as this, which will make the test solution go from blue to green. Our next test is also a contact test, which is called the catalase test, which has a test solution of hydrogen peroxide, which breaks into water and oxygen when it interacts with the catalase enzyme. The solution will appear translucent from its earlier clear color due to the oxygen bubbles that form. We chose this test because of the 3 bacteria that have hyaluronidase. Only S. aureus is catalase positive, which can help narrow down the results. It is important to also have a test that identifies if the bacteria is virulent or not. Because of this, we developed the volatile sulfur compound test, because only pathogenic strains of S. aureus release volatile sulfur compounds. It has been established that S. aureus releases dimethyl disulfide and methanethiol. Both of these compounds react with copper to make copper sulfide and copper oxide, which can turn a sheet of copper black when an infected patient blows upon it. We paired our device with the TCS 3200 color sensor with an Arduino Nano microcontroller board to detect the color changes better and more uniformly than a person's eyes could. In the finished device, we would integrate the sensor into the lining of the 3D printed device's casing, and we calculated our device to cost less than $30, so it can still be cheap enough for families in need. Also, this device would be reusable so patients could continue to use them to further increase the value of the product. Our tests were then evaluated for the best time, volume, and ratio of test solution. Each solution was placed into the device during some of the trials so that the conditions could mimic actual patients. We also took air pressure in a closed flask of bacterial broth to ensure that there was sufficient metabolites released by the bacteria. In this study, we used Enterococcus facialis as S. pyogenes and S. pneumoniae were not available, and this bacteria is a close biological relative to them. We recorded the air pressure for 5 days in the flask and found the bacteria release a significant amount of air, and the pattern followed a relatively linear fashion. We could indicate that which can indicate that the S. aureus releases metabolites at a near constant rate. We performed trials in various volumes of calcium hydroxide to find the best amount of solution to put into the device to get results in a timely manner. 2 healthy student researchers blew for each trial, and we found that 2 milliliters was the optimal amount for the device, while the patient can maintain breath speed without fatigue. You can see in the picture on the slide that is almost hot pink color, and then after this trial, you can see it's clear afterwards. The table shows the results of the hyaluronidase test. We performed our trials with both bacteria and found that the Enterococcus facialis did not react with the solution and yielded no color change. The staph cultures reacted optimally at a solute to solvent ratio of 3:1 for 6.5 seconds for a noticeable color change. On the side, you can see that it's gone from aqua blue to a lighter green. For the catalase test, the average time taken for a color change when testing with an S. aureus culture was 4.5 seconds. While there is no observed reaction when testing with E. fasciitis, the hydrogen peroxide changed from a clear liquid to a milky white when it reacted with S. aureus cultures. The hydrogen sulfide test did not result in a reaction because the tested bacterial strains were non-pathogenic. The pictures on this slide show the device design, which features a large breathing tube and hole so patients can maximize the airflow going into the device. Both parts connect to form an airtight seal to keep outside contaminants. There are 4 compartments on the bottom for each test solution to be poured into, and samples were printed to ensure quality and plausibility of such a device. Finally, we conducted 30 chest trials in the device to see any changes in the condition in the real setting that patients would be using this device. And you can see on the graph that it just shows the expected values versus our observed values and the differences. The air pressure showed that there was enough accumulation of gas produced by the bacteria to be used for detection. Fisher's exact test gave a p-value of less than 0.01 in the effectiveness of these methods, finding that this study gives preliminary confirmation of enzyme biomarkers and VOCs as potential diagnosis methods for patients with bacterial infectious diseases. I recommend for future studies to have a higher concentration of hyaluronic acid when testing the hyaluronidase test, just because the amount of acid we had was very weak, and so there could be more of a noticeable color change. Also, adding a heating coil to the device may make it slightly more expensive, but it would still keep the value low and help the Benedict solution to have a more noticeable color change with that test as well. I would also recommend more trials just so we can have better results and make sure that everything goes more smoother than just the 30 we had. And I would recommend trials with S. pathogenis and pneumonia to make sure the catalase test runs smoothly. Thank you to Divya Nori for being my partner and helping me with this event. And thanks to STEM for Social Cause for giving me a platform to talk about my research. Thank you to Berry College for giving me all the opportunities and the Governor's Honors Program.